Complicated material 101. We stopped here. It's okay. I'm interrupting myself today. Ignoring we stopped here. The whole idea of what we're going to talk about is to give you some concept of how this whole process works. So the idea is called the sliding filament theory. There's three different sections. We're going to look at next week, or Friday rather, we're going to look at how the nerve basically tells the muscle to move. Today, I'm going to show you an animation about really what happens sort of inside and how the signal gets there, because it gives you some context. And then lastly, we have to look at really the intricacy of how it slide over one another and how they release from one another. So there's a lot to this. And last time was a ton of terminology. This is a ton of really complex material, I think. It's all about these two fibers interacting with one another. This is called actin. It's the same actin that made pseudopods, the same actin that had endocytosis and exocytosis. It's the same actin that led amoeboid motion, except it's that actin you could build, assemble, and disassemble, not this. This actin is filamentous or fibrous actin. It goes by the name of F-actin, and you'll see the configuration. It looks kind of like a string of pearls that's sort of doubled over. This is the other big player. It's called myosin. And myosin's very unique, and it all has to do with how these two guys attach to one another. Because effectively, as you're going to see, the individual strands of myosin, which is the moving protein, kind of have a stem and a head that's kind of like this. And the head's kind of cocked up. It's actually in what we call the loaded or cocked position. And what happens is it has an area here that comes in contact with an area on the actin. And when the signal is given and they're allowed to come into contact with each other, it immediately fires. And effectively what it will do is it will pull the actin here and the actin here. All of these will come close to that midline. And in doing so, the more they fire, the closer they get. And in doing so, it'll bring the borders of this unique repeat unit called the sarcomere closer together. And in doing so, the filaments, because these are called filaments, microfilaments, slide over one another. And in doing so, each successive unit, sarcomere of thousands of them, shorten. And when they're shortening, effectively that causes the motion. There's no bending, there's no folding. Again, that's all about the shape of the muscle, how it actually appears to you and what it tends to be doing. But inside, and that's what we're interested in, coming together that way. So the thick filaments are made of myosin. Proteins are made of these polypeptide chains. That's what makes them proteins. Okay, and they have heavy and light ones. And so the tail, the elongated portion you will see, okay, is braided together. The heads are more globular, interestingly shaped, okay, and they're made as well. And what I was talking about is this ability to form a cross bridge. And the cross bridge is the relationship between the strands of actin and the heads of myosin. And what you'll see that as we go on, that is going to occur unless something prevents it. And really the control of whether your muscles fire or muscles don't has to do with what we call a regulatory protein that has more confusing names. Don't have to thank me, but it's coming your way. See, it was an opportunity to smile, but you're all looking at me in shock. I'm overwhelming you. And that's why I will take extra time with this. I have an idea in the second half of the class how much time we have to allot for this and how much time we have to allot for the end game, so to speak. So I mean, we're past the halfway point, so we have what to do. So we may have a couple of weeks worth of going over this one particular chapter, because it's the building chapter. Because here we begin to understand nerves and muscles, and they form these cross bridges. You're going to see down the road how they're offset. And what offset means is when you have to lift something. If I have to lift something light, it's very easy. If I have to try to lift something heavy, it's very hard. I have to basically continue to generate movement. And in doing so, it's basically, as you will see down the road, fibers temporarily attached and released, attached and released. But they're spring loaded, so if they release, doesn't it go back? It's like pulling one of these things down and releasing it. The offset means about any time we're trying to generate more power, about half of the fibers disengage and half of the fibers are still attached. That's because of this thing we call an offset. So you'll see this is part of the very interesting design of the body that allows us to generate, we have to exert ourselves forcefully in whatever activity. It could be a routine activity, or you could be in the gym working out. Just depends what you're doing. So you see that, and there's some very good graphics that will demonstrate that. You will not see those today. We will see them next week. So the thin filaments, we call it F, or fibrous actin, or filamentous actin. It doesn't matter. Okay. The actin, the ones that assemble and disassemble, are globular units called G-actin. And on those, they have sites, as you will see, for the attachment. Okay? I'm going to go over those sites in the naming in a second. They form together, but they don't disassemble. They're there. That's the thing. Muscle cells have reached the end stage of their development. So when something goes wrong, basically they're destroyed. There may be minor things that go wrong, but the reality is, by and large, they're destroyed. And so you will begin to see the way that these threads kind of twist, and that's why I like the double string of pearls analogy. And these are these very complicated names called tropomyosin. That's a thread-like regulatory protein, and it's held in place, as I will attempt to poorly demonstrate, by something called troponin, which is a plug-like protein. And those are all important. Troponin is a real important name if you're going clinical. In the old days, if somebody had a heart attack, it took us about 3 days in the hospital to make sure. We had to do serial EKGs rather, serial enzyme studies. Didn't know what the heck was happening. And all you do is worried that it might get bad. That was back 50 years ago. Now you do a blood test. It's called a troponin level. What's called cardiac troponin, because there's different types of it in different muscles. If that's in your bloodstream where it shouldn't be, guess what? There's heart muscle died, and the troponin leaks out in the bloodstream. Bang, slam dunk. Mr. X, Miss X, Mrs. X had a heart attack. So that's one of those little useful nuggets clinically, because that's a quick test that we didn't have the luxury of 50 years ago. Just give you an idea. So here's what it looks like. It's neat looking. It's got that. I apologize. I was going to buy Twizzlers. Do you know what a pack of Twizzlers costs today? $6.77. It's an outrage. My first job was in a candy store. Okay. Hershey bars were a nickel. I know. We had running water. There was electricity. I know you're surprised at that. 20 miles from New York City. We were civilized. So I deferred getting the Twizzlers, and more importantly, the peel-apart Twizzlers. So here's the stem. Here are the heads that are cocked up. That's the loaded position. Look up here. They have things called actin binding sites. They bind to the actin. This is myosin. You will recall that a lot of times we name things anatomically, not from what they're on, but where they're going. Radial head, where does it form a joint? With the radial notch on the ulna. So you can have something that says radial, but it's on a different bone. With zygomatic arch is made of the zygomatic process of the temporal bone, meaning temporal process of the zygomatic bone. Arch. That's anatomy stuff that you may or may not have emphasized. I do in my classes that are there. Okay. And this is that flexible region right now. When it's fired, it's like this. This is the cock. That means energy's been put in. We get to that toward the latter portions of this, not today, but of this unit. This is what, now notice, the same binding sites are on the actin. And if you go back, they're really just about next to each other. So once they're exposed to one another, they bond instantaneously. And so as you can see, they're called myosin binding sites. Here's the tropomyosin. It's like a thread that covers those sites. That's a bad illustration. The troponin is like a thumbtack. or a push pin, whatever you want to call it. Okay, that's holding it in place. And as you will see down the road, when calcium is let loose inside this apparatus we call the sarcomere and the muscle fiber, it alters the shape of that plug, the troponin, and it shifts out of position. I mean, realistically, here's the bad demonstration. You don't have to thank me. It's all part of the service. Here's covering. The edges are where those active sites are. And so what happens, as long as it's like this, imagine I've got the push pins in. But as soon as those push pins move or get out of shape, there they go. Now it exposes those actives. Don't thank me. It's best I could do on short notice rather than spend effectively $13.54 at Walmart. I would have eaten all the candy, believe me. I'm addicted to licorice. So you're going to see this down the road in animations. Other proteins help. Okay, we talked about Titan. That's the one that looks kind of like a coil or a spring. That's all about the elasticity that's there. Holds the thick filaments in place, helps the recoil, the stretching. They named this for the disease. When you see the prefix dis, like a disability, a lack of a certain ability, okay? Disambiguity. I was trying to be very clear rather than ambiguous. All sorts of fancy names, and that's how you can use fancy words to your advantage, okay, that are there. Dystrophin means trophy means growth. So dystrophin means it grew improperly or inadequately. And as you might have, and these are other proteins you certainly don't have to know, but the muscular dystrophy being incorrect growth, they named the protein because they didn't have a name for it that was impacted by this genetic defect as dystrophin. So sometimes we do that for convenience. Okay, you don't have to know this stuff either, but I would be familiar with this one. It's a really significant disease, even though we don't see it very much. Okay, and it's one of those nasty ones that we talk about. Genetic diseases often don't demonstrate themselves immediately in childhood, but as the child, just like Tay-Sachs, as they begin to develop, bad things. This is sex-linked recessive, and many of you have a background in genetics. We're not doing genetics here. We're not doing those neat Punnett squares and things like that. But what it means is you inherit it from the mom. So that's why it's nearly always boys. Sex-linked means it's on the X chromosome. So if you have a regular X chromosome and one with the trait, it masks it because it's a recessive trait. Boys only have Y and X, and the Y chromosome doesn't impact it at all, so it's present. That's why when you would see it always in males, okay, because it would be very rare, almost impossible to imagine in females just because they don't live very long. And so it generally starts about then, and what happens is this particular thing doesn't allow the muscle to grow and develop properly. It deteriorates, it's replaced by fatty tissue and connective tissue, and eventually what happens with a lot of muscular diseases, respiration. Eventually the child, when they get probably between 30 and 40 traditionally, they begin to succumb to respiratory chronic pneumonia problems, heart problems because it affects heart muscles, and the lifespan was very limited. That's what they're telling you here. So, and this is about 8 years ago. The longer we work on these diseases, the improvements are dramatic in how much they are. So this is the defective gene called dystrophin, okay, and it tells you what it does, and the sarcolemma tears, inflammation, regenerative capacity is lost, blah, 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 blah. And it goes, there's a picture, and they're just telling you all those things. I made this side note to the other classes, so I'll make it to you. Years ago, even though it was a disease that did affect a number of people tragically, not very many. So what changed medicine a bit, and particularly big pharmaceutical companies. Big pharmaceutical company, the answer is in the last word, company. Companies are about making money for stockholders and themselves. Okay, the fact that they make pharmaceuticals doesn't make them terribly nice. It's the realities of the world. So they didn't have an incentive to do research or fund research to find out how to treat this. When AIDS became very, very noticeable and very prominent people began to succumb to it, and we began to learn about HIV, and I go over this when we do micro together and more thoroughly. Congress, at the urging mostly of entertainment and Hollywood people, because they had a lot of money and a lot of influence, passed something called the Orphan Drug Act in 1988. It has nothing to do with orphans. The orphan concept was to fund research, development, and fast track it for diseases that don't affect a lot of people. Read that, HIV/AIDS. And so also these other things, that's why we're seeing more research. Amyotrophic lateral sclerosis, Lou Gehrig's disease, would be another example. Ultimately, it comes down to the funding and the money, and whether in big companies, they need to have incentive to do things which are not ordinarily going to be profitable. That's just be realistic. Because you get into that stage in your lives where you have to think in that context. Very, very much so. There's a certain loss of naivete as we get older. I'm still learning about that, and I'm going to be 75 at my next birthday. I know you're all looking at me going, that's impossible. Those of you who are awake. You could have smiled and said, no, you couldn't be. I need someone who's a barometer. So, the sarcoplasmic reticulum, you will see this in the animation. This is a unique type of smooth endoplasmic reticulum. They don't need rough because they've already made all their proteins. The smooth endoplasmic reticulum contains calcium. It's got little wells called cisterns, and they store and release calcium. It's all about the calcium that's in the cell. We call it intracellular calcium. When we talk about the heart, cardiac muscle, and smooth muscle, that's calcium in your bloodstream. So calcium, this is really the introduction of why calcium is so darn important. And indeed, for the nerve to fire, to tell that muscle to fire, it requires calcium in your bloodstream. But the calcium is already in the muscle cells and kind of stays there. That will be a distinction we will look at as well. They happen to be, as you will see from the illustration and from the animation along the A and I band. I'm sure, I'll try to make sure it's not a big deal on the test for us. I usually ask my advanced students about that. And really, it's all about regulating the release. The release is triggered by a very interesting, I would use the term invagination. It's like a folding of the sarcolemma or the membrane. They call it T or transverse tubules, as you will see when I show you the animation, because they go at right angles. Normally, the membrane follows the long axis of any cell. These go as well along this way, tangentially or at right angles. And so they are able to send the impulse of the nerve down deep along all those neat fibrils, and basically send the signal for the filaments to slide over one another. So they penetrate deeply, and they form something called a triad. And the triad definition is one of these transverse tubules, membrane, with sarcoplasmic reticulum wells called terminal cisterns on either side. And I'll show you the animation now so you get an idea. And what's going to happen, they will show you. What allows the calcium to be released is that signal travels rapidly down that cistern. Okay, I beg your pardon, rapidly down that transverse tubule. It triggers a little voltage sensor, like a light switch, and snaps it. And when that snaps, it allows the cistern to open its doors and allow calcium to flood the cells. I will jump ahead, and you'll see a whole bunch of stuff that we're stuck talking about. This is the degree of complexity, everybody, that you're going to get. Look at all that. That's coming up next time. And there's animations about all of it. We're coming to one, and I'll make me look bad. A lot of good stuff there. Gotcha.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with a calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
And I mean, we're going to view this again when we really look more deeply at that cycle. But it's hard to describe how extensive and how interrelated the sarcoplasmic reticulum and these guys, which are the transverse tubules, what they're surrounding is each of the fibrils. The fiber is the cell, and it's got those flexible tubes called fibrils, and the filaments, the actin and myosin, are inside. So here you see them, and they're evenly spaced. Happens to be along the A and I band junction, but we won't go there for now. And effectively, what happens, they are associated with this extensive network of endoplasmic reticulum, which is smooth. And one of their jobs of smooth endoplasmic reticulum is to store calcium when necessary. This is a triad. So in the dark and the blue on either side, that is the cisterns, these enlargements, which they call wells, but that's fine. And then you can see what happens when you see that light, that indicates that an energy bolt is going through, tripping that voltage gated sensor or channel. And that trips it in such a way that, and you can see it's married on either side effectively to a gateway that allows calcium to pour out of and into the interior of the cell. That calcium, ladies and gentlemen, is going to bond to the troponin, those plugs of protein. Anything that attaches to a protein changes its shape. The shape will change, and voila, without having to do that with the cord again, it's going to basically expose those areas. And then once they're exposed, contraction begins. The longer they're exposed, the more calcium is out there, because it'll keep resetting, fire, reset, and it'll get stronger and stronger and stronger, just like you have to go out, you have a big heavy package that came from Amazon, and you got to lift it up. That's all. It's kind of the way that whole thing works. So I'm going to go backwards and go back to where we were and try to give you some context before we do. And you can see, look at the stuff that's going on here. We're going to have a lot of talking about the steps that involve the nerve. These are just some of the steps we call the events at the neuromuscular junction. And it's so pivotal because our next chapter is all about nervous system physiology. We're going to look at what nerves are made of on a microscopic level. We're going to look at the parts of the brain. And more importantly, we're going to look at how basically communications occur. You're being able to tell muscles, your brain having to fire, your brain processing information. It's extraordinarily important. And so this kind of stuff is really what you're sort of getting the framework. And that's the beauty of these two chapters, is they're interlinked together. So, I mean, there's a lot here. I can't say it strongly enough. I sent everybody a message I contacted the tutoring center at the Center for Student Success. I strongly recommend using their resources. I really do. Listen to these things. There's nobody's listening to my recordings, but I'm going to keep doing them. I mean, I hate to put it this way, but if the departments come to me, what happened with all your students? I'm going to say they don't listen. Please listen. You don't believe this. Your success is my success. It really is. I don't got a whole lot else to do. I'm going to retire in a couple of years. I'll be fine. You're getting started. So I like to hear from them say, "Oh, I just got this. I got my BSN. I got my MSN. I've got my own physical therapy practice. Whatever." That's the part we take home, whether it's doctors or nurses or whatever. And I'm being very sincere with you. So this is what we're talking about in earnest here, and this is where we kind of left it with these T-tubules and what they call triad relationships. So, and really that's the important part, to understand what we're talking about today. So the sarcoplasmic reticulum or cistern, and then we're talking about the integral proteins, and then eventually the relationships and the proteins changing shape. Proteins change shape, two types. There are ones we call ligand or chemically gated channels. The terms are interchangeable. And we have voltage gated channels. And voltage, because excitable cells, which nerve and muscle cells are by definition, every cell has a resting membrane potentials. What it means to be excitable, it means that potential along the membrane can change rapidly. Again, like flipping a switch, the lights go on immediately, but imagine that wave moving, and basically it's an oscillating wave. Ripples in the water, whatever works for you, doesn't matter. Effectively, to understand, it's a rapid change of the polarity going from plus on the outside, minus on the inside, flipping and flipping back, a la the switch. And as it does that, that wave of energy goes by and opens that channel. And you're going to be even more amazed. The calcium outside in the bloodstream has another channel that opens. And when that wave comes down the nerve to either the next nerve or the muscle, we have another voltage. We go from voltage gated to ligand gated to ligand gated to voltage gated. You're going to get sick and tired of it. It is very complicated. So this is like I said, this is the hard stuff. We did the easy stuff, which will give you some context, because that was not necessarily easy. It's just comparison. Here's what it looks like again. And you can see it's investing. Let me go back. All of these are the fibrils, the little blue and red guys. Those are the filaments, the actin and myosin. Contraction, okay, shortening, okay, and basically the contraction ends as the cross bridges, when the cross bridges become inactive. In other words, you'll see, we'll get into what stops it. And interestingly, to make it go back to relax, we have to put energy into it at the end. There's a lot of energy that's involved. Energy to fire the nerve, energy to fire the muscle, energy to get the muscle to relax, energy to pump the calcium back in so it can be used again. A lot of energy. So the thin and thick filaments overlap. We call it the sliding filament. During contraction, the thin filaments slide past, okay, causing the actin and myosin to overlap. They don't change length. They don't change shape. The illusion is because of the overall anatomy of the muscle. Again, all those individual sarcomeres, depending, whether it's pennate, convergent, who cares? All those good terms you learn in anatomy. Basically, that how the muscle, what the muscle, oh, it's contracting, it's folding. No, it just has to do with the arrangement that are there. And so when the nervous system says, grab this, okay, that muscle fiber, or whichever fibers, and it's more than one that you're activating, the myosin heads are allowed to bind to the actin, thank you, calcium, cross bridge, and then eventually you're going to see that sliding process. And effectively, what happens, to summarize this, the Z discs are pulled close to the midline. And you're going to see this, and particularly the I band shortening, and the Z discs appearing close to the other. I don't really care about that stuff. So here's what it looks like micrographically. Here's that same, more or less, illustration. The amount of excursion that's available, How far it can travel is specifically how this blue area, that's the tip of the axon, can get all the way to the midline on either side. That's maximum contracted. You're going to see maximum contracted. How it's going to be visibly changed, this is going to look a little different in the middle. These are going to be cut in half, more or less. So here's the cursor, here's the change. Notice that's the I band, the light zone. It's tiny. But look what happens if I go back to where it was initially. Wide on both sides. So effectively, if you look at the Z disc on either side, they're moving equivalently toward the midline. So as we go forward, now they're closer together. Here it is here. This is where it started. So if I back up, here's the cursor. When I go forward, now it's moved in almost half of that white band. That's maximal contraction. That's what's going on repetitively. The endpoint is met here. Why they say fully contracted, and effectively the schematic aspect of it is the axons have moved as far as they can go. There's nothing, they're physiologically unable. You can't, per se, overflex the muscle in that regard. I suppose you could try, but there would be damage that was associated with it. You could try to mechanically force it, but I don't really think so. So that's really what I need you to understand mostly for today is the intricacy of this process. But really, in order for us to really step by step to understand this whole idea, there's three major phases. The middle one you saw, but we'll go over it again. The first phase is called the events at the neuromuscular junction. Read that as your brain telling this to move. It is a two-nerve circuit. We go over this again when we get to the brain and parts of the brain and the spinal cord. Basically, from your brain to your spinal cord, let's say I'm going to move my arm, it goes from my brain to my spinal cord, roughly in this area, in the brachial area, and then it shoots out to my arm so I can grab something. It's more than one, obviously. But the nerves talk to each other by releasing chemicals. That's how nerves work. It's not an electrical thing like putting a plug in the wall and it's an extension cord. I'm going to put another plug in. That's electric to electric. It's very, very rapid. They talk to each other by chemicals. The chemicals are called neurotransmitters. The one we learn about in this chapter, thankfully, is the one that you need to know the most about. It's the one that you have the most of in your body. It's in your brain, every neuromuscular junction for skeletal muscle. It's in what's called the autonomic nervous system, which we do at the very end of the course. All the things that control your body without you having to think about it. And acetylcholine plays a role in all of these. It's extraordinarily important. Your heart rate, acetylcholine. Keeps your heart rate in check. You did cranial nerves, right? Remember the vagus, the 10th cranial nerve, ringing a bell? Yes. Longest cranial nerve, only one that leaves the head and neck area, innervates the heart. It releases acetylcholine. The old name for it was vagus stuffle. Stuff out of the vagus. Except we know it's made from an extract of acetic acid, that's vinegar, and something called choline, which we use a lot in the body. So the decision to move is activated by your brain, and the signal is transmitted first down the spinal cord to whatever level it's supposed to be in, and it junks with another nerve in the spinal cord and goes out to the muscle to say, move it. Okay, nerves and muscle cells are excitable cells. That means that can rapidly change that resting membrane potential and rapidly reset it. So they change it and go back to the original thing, the action potential. And action potential is a unique word. It means literally the potential of action or to do something. The fact that an action potential isn't always generated. Think of you deciding to do something. Maybe you're going to, should I watch this program on TV or listen to something I'm listening to, let's say on YouTube arbitrarily, or should I go and study? I mean, the action potential is when you make a decision to do one or the other. Thinking about it is kind of like not an action potential. You may be, nah, I don't feel like doing that yet. It's like me recording those review sessions, but I do them for you because it's my job. You get the idea. I know. Tell my wife. They're listening to their grandpa telling him this stuff. It doesn't come in. It's all right. My grandchildren don't listen to me. So what happens is it's all about charged particles moving in and out, as you will see. Those channels, when nerve and muscles function, are either ligand or chemically, same word, gated ion channels opened by chemicals like neurotransmitters, or there's the acetylcholine, that's the abbreviation, or voltage gated ion channels, which is all about that electrical current, the switch. And this is what they look like. This is acetylcholine. It's bonding to this, basically a protein, and it allows that protein to open and close, just like I did with the stupid folding it and rolling it and they open up and close. So I did that. When something attaches to its protein, it changes shape, opens or closed. And this is what happens with voltage. That little guy we'll get to. That's called an inactivation gate. And it really isn't that relevant to us now. It gets a lot more relevant when you do heart. And interesting, if these are gates, this is the lock. Every so often you have to lock one of those gates. That's what an activation gate is. Don't have to worry about that now. It's coming. So the anatomy of nerves and the neuromuscular junction is what we're going to look at going forward. So we're effectively done. But I did want to show you, I mean, there's a lot of these are all cells, and you're going to see down the road that we activate. For us to do that one thing, we activate different areas along that muscle. Why do we activate different areas? So one doesn't fatigue. We're going to look at all that goes on there. We're going to look at the junction itself. We're going to look at this, which is called the motor end plate. And so we're going to first look at this and all the events there. Then basically review that video, but in detail, call it excitation, contraction, coupling. And really, it's a lot more sophisticated than what you saw there. And then finally, the actual way that the contraction occurs and how it relaxes and how it resets itself, called cross-bridge cycling. And that's really today's presentation. I know it's overwhelming, so I didn't want to go much longer and more detailed than this. Thank you very much. Don't want to give you the old information overload.